Okay, so welcome to this next video in which we are discussing chronic gastritis and peptic ulcers. Okay, right, so one more piece of physiology. What we want to discuss is how the cells of the gastric mucosa secrete prostaglandin E2 and the protective role that prostaglandin E2 has on the gastric mucosa in protecting it from the corrosiveness of the extreme acidity of the contents of the stomach lumen. Okay, so basically, prostaglandin E2 is synthesized from arachidonic acid. So we'll start with arachidonic acid. Okay, so arachidonic acid can be converted firstly into prostaglandin G2, and then it can go on further to prostaglandin H2. Okay, and these conversions are going to be catalyzed by cyclooxygenase enzymes. Now, prostaglandin G2 for short is often abbreviated to PGG2. Okay, and then prostaglandin G2, as I said, can be converted further to prostaglandin H2, and I won't write its name out in full, I'll just abbreviate it to PG for prostaglandin and then H2. Okay, so these two reactions are so important that they even have names. The conversion of arachidonic acid into prostaglandin G2 is called the cyclooxygenase reaction. Okay, and then the conversion of prostaglandin G2 into prostaglandin H2 is then called the peroxidase reaction. Now, both the cyclooxygenase reaction and then the peroxidase reaction are catalyzed by the cyclooxygenase enzymes, of which there are two main forms, and potentially there's a third form as well. But for our purposes, we're only going to discuss the two uncontroversial forms. Okay, so there is cyclooxygenase 1 and there is cyclooxygenase 2. And cyclooxygenase is usually abbreviated just to COX. C for cyclo, ox for oxygenase. So you have cyclooxygenase 1 or COX1, and then you have cyclooxygenase 2 or COX2. Okay, so both of these enzymes catalyze this conversion of arachidonic acid firstly into prostaglandin G2 and then on to prostaglandin H2. Now, what do you then do with prostaglandin H2? Well, you can convert it into a huge number of things, but one of the main things that it's being converted to by cells in the gastric mucosa is prostaglandin E2. Okay, and this is done by the enzyme prostaglandin E2 synthase. Okay, and then the prostaglandin E2 will then be secreted from the cell. So within the gastric mucosa, within the lamina propria, you have a lot of prostaglandin E2 just floating around. Now, what does this do? Well, basically, it's going to act on both the columnar epithelial cells, which remember secrete bicarbonate and mucus, and it's also going to act on the enterochromaffin-like cells, which remember control the secretion of hydrochloric acid by the parietal cells. And basically, prostaglandin E2 takes measures to protect the columnar epithelial cells from the corrosive power of the hydrochloric acid. So it's going to increase the secretion of bicarbonate, anions, and mucus by the columnar epithelial cells. So it's going to increase the protection that the columnar epithelial cells have against the hydrochloric acid, and it's also going to decrease the secretion of hydrochloric acid by decreasing the secretion of histamine by the enterochromaffin-like cells, which remember is directly proportional to the amount of hydrochloric acid that is then secreted by the parietal cells. So it's going to inhibit the secretion of hydrochloric acid by stopping the enterochromaffin-like cells secreting the histamine, which stimulates the secretion of hydrochloric acid by the parietal cells. Okay, so let's now talk about the receptors that it acts on to achieve all of these things. So basically, on the surface of the columnar epithelial cells, you have the prostaglandin E2 receptor type 4. Okay, so where should I draw this? So let's say this is a columnar epithelial cell. So basically you have four major different types of receptors to prostaglandin E2. So before we discuss the specifics, let's talk about these four receptors in general. Okay, so the 4 receptors for prostaglandin E2, they're firstly all G protein coupled receptors, and again, they are rhodopsin family G protein coupled receptors. Okay, so they bind their ligand in transmembrane domains. Most G protein coupled receptors are rhodopsin family. Okay, now there are 4 different prostaglandin E2 receptors, and they're given the rather silly names EP1, which is back to front. Basically, you put the E first, and then this P stands for prostaglandin. So this means prostaglandin E2 receptor type 1, even though it seems back to front. Okay, then you have the EP2 receptor, so at least there's some consistency. So prostaglandin E2 receptor type 2. Then there is EP3, the prostaglandin E2 receptor type 3, and then finally there is EP4, prostaglandin E2 receptor type 4. Okay, now we're going to see each of these types of receptors. And now basically on the columnar epithelial cells you have EP4, the prostaglandin E2 receptor type 4. Okay, so here is EP4. And the prostaglandin E2 will come and bind to this receptor, and again, because it's a, oops, what am I doing now? You see, the silly naming of this is getting me confused. Prostaglandin E2. Okay, the prostaglandin E2 will come and bind to this EP4 receptor, and what that triggers is it triggers an increase in the secretion of mucus by these columnar epithelial cells. Now, also on the surface of the columnar epithelial cells, you will also have the prostaglandin E2 receptor type 1s and also the prostaglandin E2 receptor type 2s. Okay, so this could either be an EP1 or an EP2 because they're going to do the same thing. Okay, and what will happen is this prostaglandin E2 could bind to one of these receptors, and this will then increase the secretion of bicarbonate anions onto the surface of the columnar epithelial cells. So the prostaglandin E2 receptor subtype 4 triggers the increase in the secretion of mucus. Both the prostaglandin E2 receptor type 1 and the prostaglandin E2 receptor type 2 trigger an increase in the secretion of bicarbonate into the mucus to neutralize the free protons. Okay, now that's the actions of prostaglandin E2 on the columnar epithelial cells, and basically it's saying put your shields up, protect yourself, okay, so double the protective forces. Right, prostaglandin E2 also weakens the attack, however, so it also acts on the enterochromaffin-like cells, and on the enterochromaffin-like cells there are 2 types of prostaglandin E2 receptor. Okay, and they both again do the same thing. So there is the EP2 receptor, the prostaglandin E2 receptor subtype 2, and there's also the EP3 receptor, the prostaglandin E2 receptor subtype 3. And basically what these 2 receptors do when prostaglandin E2 binds to the receptor is they trigger a decrease in the secretion of histamine. So the amount of histamine that the enterochromaffin-like cell is secreting will be reduced, and this means that the parietal cells will be receiving less stimulation from the enterochromaffin-like cells, and therefore will reduce their secretion of hydrochloric acid. So not only do you increase the protection by increasing the mucus secretion and the bicarbonate secretion, but you also decrease the amount of acid that's being secreted, and therefore you decrease the attack. Okay, right, so that's just a protective mechanism. Right, so what we now want to discuss is chronic gastritis. Okay, so we're now finally moving on to the pathology. So, basically, chronic gastritis precedes peptic ulcers. Okay, so you'll have chronic gastritis usually before you get a peptic ulcer. Right, so what is the cause of chronic gastritis? Well, actually, firstly, I should tell you what it is. Anything ending in itis means inflammation. So this is inflammation of the stomach. Now, specifically, it's inflammation of the stomach wall, and chronic means it's going to last for a very long time. So we've just got long-term inflammation of the stomach wall. Now, there are many different things that can cause long-term inflammation of the stomach wall. For instance, you could have an autoimmune reaction against the stomach wall, and that will cause a long-term inflammatory response against the stomach wall. However, the most common form of long-term inflammation of the stomach wall is now acknowledged to be infection by the bacterium Helicobacter pylori, and it is this form of chronic gastritis that we will focus on in this video. Okay, so long-term infection with this bacterium Helicobacter pylori. Okay, so firstly, let's draw a picture of Helicobacter pylori, and then let's discuss where it's going to live, and we've already mentioned this. Okay, so basically, Helicobacter pylori is this long, rod-shaped bacterium that sort of curves around in an alpha helical sort of shape, like so. And then off the edge of it, it has many flagella, basically, so I'll show these here. So here are its flagella, okay, which it uses to propel itself along, okay, and then its body is this long rod, so it's a bacillus, and it's basically gram-negative, okay, so if you gram stain it, it will appear red down the microscope. Now, what does that mean functionally? Well, it means that it has two lipid bilayers. It has a cell membrane and then also an outer membrane, and it has a very thin cell wall that sits in between those two cell membranes within the periplasmic space. Okay, so I'll colour it in red since it's gram-negative. Okay, and the reason it then appears red in the gram staining process is that it doesn't hold on to the initial crystal violet stain. So in the gram staining process, what you do is you first put on a blue stain onto all the bacteria. So if you've got a little plate of bacteria that you want to stain, what you'll do is you'll put on a blue stain, which is called the crystal violet stain. Okay, and this binds to the cell wall of the bacterium. Okay, now in in gram-positive bacterium, we'll say first. Gram-positive bacteria only have one cell membrane, so here's the cell membrane, and then surrounding that cell membrane, they have a very, very thick cell wall, okay, which I'll show in green here. So big, thick peptidoglycan cell wall, okay? Whereas gram-negative bacterium, they have two cell membranes, like so. A cell membrane, which is the inner one, and then an outer membrane. But then they have a very thin cell wall in between these two membranes here. And basically, what you then do is you apply iodine. After you've put the crystal violet on, you apply iodine, and basically this fixes the crystal violet stain, this blue stain, and I'll underline it in blue to highlight that it's a blue stain. It fixes it to the cell wall. Now, in the bacterium which have extremely thick cell walls, the crystal violet will get fixed to that extremely thick cell wall. So the gram-positive bacterium will get loads of this crystal violet stain all stuck to them, basically, whereas the gram-negative bacteria don't get the crystal violet stain stuck to them. Okay, what then happens is you wash the bacterial plate off with a powerful solvent, such as acetone. This wipes away all of the stain molecules which are not attached onto your bacteria. Okay, so now the only crystal violet stain molecules that remain on the plate after you have washed it off with acetone are those which are bound to the peptidoglycan cell membrane. Okay, so they're bound to the gram-positive bacterium, and the gram-negative have none. Okay, what you then do is you counter-stain. You put on another stain, which is a red stain called safranin. I'm trying to work out if it's an A there or an I. Safranin. No, that's not right. Safranin. It's either an... Well, I can say it even if I can't write it. It's safranin. Okay, and it's a red stain. Right, so basically this restains all of the bacteria again. So it stains the gram-positive bacterium and the gram-negative bacterium, but it's a weaker colour than the crystal violet. So for the gram-positive bacterium, which are covered in this gram-positive crystal violet stain, you just cannot see the safranin. Okay, the crystal violet stain is too powerful colourfully that you just can't see the redness coming from the safranin. So they appear blue, basically, the colour of the crystal violet stain. But for the gram-negative bacterium, they don't have any crystal violet stain. Okay, so the only stain that you see is the safranin stain, and that appears red, so you see gram-negative bacterium as red. Okay, so that's just a little review of the gram staining process. So Helicobacter pylori is a gram-negative bacterium. It appears red if you stain it with this process. Okay, so enough of that. Let's now discuss what Helicobacter pylori does. So basically this bacterium is capable of colonizing the surface of the stomach. So it lives on the surface of the stomach. So let me draw a picture of the stomach. So here is the stomach, and it will be living on the surface. Now, you might ask, well, how on earth can it live on the surface of the stomach? There's the hydrochloric acid within the stomach that's supposed to stop bacteria from being able to live in there. But basically, it lives within the mucus that covers the epithelial cells. Okay, so here we have these columnar epithelial cells lining the surface of the stomach. And basically we know that the columnar epithelial cells secrete this lovely thick layer of mucus to protect themselves. And they secrete bicarbonate anions into this mucus so that the pH of it is kept around 6 to 7. Okay, and it is in here that the Helicobacter pylori bacterium will live. Okay, so you end up with a whole colony of Helicobacter pylori cells living in your mucus that lines the surface of your stomach, basically. Now, basically, they are brilliantly designed to live in this mucus. Firstly, they have these flagella, which allow them to propel themselves through the thick mucus. Secondly, they also help keep the pH a desirable level. So the mucosal cells are doing their best. They are secreting the bicarbonate, which helps, but the Helicobacter pylori isn't satisfied with the good work of the mucosal epithelial cells. It does the job better, it thinks. So what it creates is an enzyme called urease, okay, which is going to break down urea into ammonia. And ammonia is a strong base, so will also absorb protons, basically. So it's going to help the bicarbonate anions, and it's going to help the Helicobacter pylori maintain a nice, comfortable pH for it to live in. Okay, so let me just show you this reaction. So the structure of urea is basically like so. You have a carbonyl group in the center with 2 amino groups coming off this carbonyl group. Okay, and basically what you're going to do is you're going to use 2 water molecules, okay? Oh, sorry, no, not 2 water molecules, 1 water molecule to break this structure apart, okay? And if I just draw the water molecule as well, so you have an oxygen here with 2 protons coming off it, okay? And basically what you're going to do is, well, what you can imagine doing. Again, this isn't an electronic flow diagram, this is just how to make sense of the reaction. What you can imagine doing is breaking these 2 bonds here, and you can imagine giving 1 electron back to the carbon and 1 back to the nitrogen in each case here. Okay, that leaves this carbon with 2 free electrons and each of these nitrogens with 1 free electron. Then you can imagine doing the same thing to these bonds between the oxygen and the hydrogens within water. Okay, so give 1 electron back to the oxygens, to the oxygen rather, and 1 electron back to the hydrogens. Okay, so that will leave this oxygen with 2 free electrons and each of these hydrogens with 1 free electron. And you can see what's going to happen now. Bind this carbon which has 2 free electrons to this oxygen which has 2 free electrons to create a double bond between that carbon and that oxygen, and therefore you've created a molecule of carbon dioxide here. okay, then bind the 2 protons onto each of these 2 nitrogen atoms to create 2 ammonia molecules. Okay, so ammonia is NH3, so a nitrogen atom with 3 protons, well, 3 hydrogen atoms covalently bound to it. Now, the important thing to understand about ammonia is that it has a lone pair of electrons, which acts as a center of negative charge. So, if you have 3 protons within the medium, then basically they can latch onto the lone pair of electrons, and basically this is how ammonia molecules can accept protons on to create ammonium cations, NH4+. Okay, and this will mop up free electrons. So basically, the urease created by the Helicobacter pylori is going to synthesize more base molecules, which will help to neutralize the pH within the mucus where the Helicobacter pylori is trying to live. Okay, right. Other things which help them to live in this mucus is that they have special molecules called adhesins, okay, which are little proteinaceous extensions from their membrane, and basically what they will do is they will stick this protein extension into the columnar epithelial cells, and they will use it to latch themselves onto the surface of the stomach, basically. So this is what is known as an adhesin. Okay. Finally, they also secrete a toxin, okay? And this toxin is known as CAG-A, okay? And CAG-A stands for the cytotoxin-associated gene A. So C is for cytotoxin, the A is for associated, okay, and there's a little dash between cytotoxin and associated. The G is for gene, and then the A is for A. So this is the cytotoxin-associated gene A. Now, this cytotoxin-associated gene A, it's not fully understood what this does yet, but it seems to be involved in the formation of peptic ulcers. Okay. Right, so there's our introduction to Helicobacter pylori complete. In the next video, what we'll discuss is what the stomach's response to having its mucus colonized with these Helicobacter pylori bacteria is, and how the stomach's response then leads to the formation of peptic ulcers, because basically the stomach's response to this is, well, how are we going to eradicate this? Well, firstly, we'll bring in the immune system, but also, how do we usually eradicate bacteria from the stomach? We usually use the hydrochloric acid. So basically, the stomach's response is, well, let's secrete more hydrochloric acid, let's burn these bacteria out. Okay, but of course what ends up happening is that it ends up burning holes in its wall, basically, and that's a peptic ulcer. And the problem is that unlike acute gastric ulcers where you get burning of the mucosal layer, acute gastric ulcers are acute, okay? So the cause of the burning of the mucosal layer of the stomach wall is only temporary, okay, and it heals very quickly, whereas basically the stomach fails to clear the Helicobacter pylori but doesn't give in trying, so it continues to produce excessive hydrochloric acid for extremely long periods of time, and then you get these chronic wounds to the side of the stomach because of too much acid being produced within the stomach. Okay, and we'll go over that again in the next video.